Sure. So what sort of broadly has been looked at in context of HBV or B virus and the risk of liver cancer has been really been related back to the effect of the virus in induction of chronic inflammation and that sort of producing or promoting cancer development. But it's been known, obviously, that many individuals carry this virus, especially childhood acquisition of the HBV, hepatitis B virus, and they go on asymptomatically for life. And having the virus doesn't really equal getting into trouble with cancers and chronic inflammation. So we really wanted to understand what is the mechanism linking the two, in fact, what is the mechanism that could be uh, allowing or promoting cancer development in HBV positive liver. And what we found was that HBV presence of the genome itself is not sufficient to induce chronic inflammation or cancer. It's really the exposure of that liver down the road to carcinogens, to bona fide carcinogen, is what really drives chronic inflammation development and cancer development. And it seems like main thing that HBV is doing in the liver is to modify how the liver responds to environmental carcinogens and how the inflammation gets changed when that liver has HBV infection in it. So that's really the finding, which is it really brings up this hope that you could prevent cancer development and chronic inflammation in patients who are HBV positive by preventing the exposure to those carcinogens or reducing the risk of exposure to various carcinogens that are in our environment. The reason we use that chemical, which is a short form of this DEN, is because of its common use actually in experimental models to induce liver cancer. But it is a very representative chemical exposure as we see in humans. Of course, the human exposures could be very broad from alcohol use to smoking to other carcinogen exposures. But this chemical is a fair representation of exposures that humans can come to as they go along their life. So that's really, again, it's if you will, a chemical that allows you to model that exposure in animal models. And that's what we have used. Yeah. Correct. So what we did in that context was obviously data sets that are very, very big data sets, and looking at use of pitavastatin, a type of lipophilic statin that we previously shown can block the cytokine IL-33, which we think is a key molecule intermediate between the carcinogen HBV exposure and cancer development, chronic inflammation development. So that was the statin group, and then we used another cholesterol-lowering drug that is not interacting in the same mechanism as the comparator group to match the groups and be able to see this risk reduction in the statin use in patients. We control, of course, for age and gender and some of the ethnicities and backgrounds that we could extract from those large data sets. And that's really the population we have compared, thousands of them. I believe there's 242,000 patients in each group of these patients who received the statin versus the comparator drug that is, again, cholesterol lowering drug, but doesn't affect the same mechanism pathway for its activity. What we found, but this is a work we have done previously in our lab as a lab that is really interested in cancer prevention. You always look for agents that are safe and you can have patients use and hope that they could, mitigate the risk of cancer in them. And the statins was discovered actually after a FDA-approved drug screen. And what we found was that statin, especially pitavastatin, which is lipophilic statin, it blocks the expression of the cytokine IL-33. And as you just mentioned, IL-33 increase would stimulate regulatory T cells, then promoting that cancer phenotype and chronic inflammation in the liver. So when you can block it actually by blocking the expression of this cytokine using statins that upstream blocks the expression of the gene in the cells. And you mentioned which statins. Again, these are lipophilic statins, mostly what we have tested to be effective. Pitavastatin seem to be at least in our assays, the best performing one, but other lipophilic statins And I should say broadly, statins, even the hydrophobic ones, should have efficacy. Bioavailability of the lipophilic ones are better for the purposes that we have been testing them. Yeah. I believe the concept really relies on how safe, I guess, the intervention would be. Because again, we are looking at a large population of individuals who are carriers of the virus, but at the time that we would like to intervene and block inflammation in their liver and also cancer in their livers, they're technically healthy individuals. So that agent should be also carrying a very high profile of safety, which that is exactly the reason we focus on statins for this purpose, because they have a very nice and established safety profile in human population. So it's a type of drug that you can repurpose for prevention in this context, alone or as you just said, in combination with antiviral therapies. Now, novel treatments in terms of IL-33 blockers, those are again being developed in various formats, with from antibodies to small molecules. We are advocates of small molecule inhibitors of this cytokine because it not only has cytokine function, but it has intracellular function. So if you want to really block it, you want to block the expression of the actual protein, which is what statin does. It's a very nice molecule in that way, that it blocks complete expression of the cytokine in the liver cells. And that allows hopefully for a more effective persistence of the effect and prevention of chronic inflammation and cancer. Yeah, so the biomarker, as you just mentioned directly, is they're looking at, for example, the cytokine levels, the IL-33 levels, either in the serum or in the, if they're having some liver symptoms of chronic inflammation that warrants biopsies, looking at those expressions in that context could stratify patients at high risk versus not. And the next steps for us is really taking this much more in mechanistic direction of understanding the cellular sources of IL-33, exactly what other cells in the liver respond to this cytokine, and how can we really, as you just said, come up with perhaps more effective, even preventive approaches to enable a reduction of this risk by mitigating the effect of carcinogens during that exposure time, so that you don't allow the carriers to really have to worry about chronic inflammation and cancer. So we really need to understand better the mechanism that is going on in this context to come up with earlier therapeutics that are also safe and effective.